Okay, can I start it? Good evening, everyone. My name is Paul Chang. I'm today's speaker. And the paper I'm going to talk about today is published on the Octa Neuropathologica. And the title is the frontotemporal dementia caused by the CAM2B mutation. It's characterized the neuronal lysosomal storage pathology. And in the very beginning, I'd like to show a very astonishing statistic. The population of the dementia is keep increased from 2010 to 2015. So it would be a very crucial problem for us to face in the future. And this is why I chose this paper to present today. So as for my today's outline, first I will introduce about what is the FTD. And then I will go through the background of the article to make you have a further understanding. And the most important one is I will talk about what makes this article so special than the previous one and show several results to support that. And then I will throw some discussion and sum up my presentation. And that is the Q&A section. So let's start from the first part. What is the frontotemporal dementia? In many kinds of dementia, they can be categorized into three categories, which is the Alzheimer's disease, vascular dementia, and other dementia. And the frontotemporal dementia is in this 13%. And as this picture, you can see that the pathological area of the frontotemporal dementia are mainly at the frontal and temporal lobe, which is different from the Alzheimer's diseases. And let me tell you some facts about the FTD. The first one is quite straightforward, because the neuronal loss usually happens in the frontal and then temporal lobe. And also it is a genetic inheritance disease, which means that the patient will pass on for several generations. And the third one is compared with the different kind of dementia, FTD mostly happen in the younger generation. And because the frontal and temporal lobe are usually take charge of the behavior and the emotional control, so the FTD patient usually suffer from the emotional disabilities and the speech and the language disorders. And now let me go through the background of these articles. As you can see, there are lots of genetic mutations that can be linked to the frontotemporal dementia. Some of them will cause the toxic aggregation, and some of them will cause the abnormal cell division. But today's our main character will be put on the CAM2B mutation here. So what is the CAM2B mutation? Which is the abbreviation of the charged multivascular body protein 2B, which is the subunit of the SCAR3. The SCAR3 is usually located on the membrane, and assembling from the SCAR0 to SCAR3, it will form a complex, and then it will trigger the cells to undergo the membrane budding and bending. Usually happens in the cell divisions and the intracellular sorting process. For example, like the endosomal system, here is the early endosome, and it will be processed to the late endosome and then sent to the lysosomal process for the protein degradation. So if we created the truncated CAM2B mutant, loss of function mutant, we will affect the maturation of both endosomal and autophagosome process, and eventually affect the lysosomal degradation. In previous research, the scientists have found that in mice, mutant with CAM2B mutation will show the overgrowth of the microglia cells and some P62 inclusion, which is frequently found in several kinds of dementia diseases. And now I'm going to go into the most important part, is what makes this article so special. Some clinical doctors have found that the FTD patient with the CAM2B truncated mutant can be observed lots of autofluorescent aggregates in their frontal lobe. The autofluorescent aggregate is different from the lipofuscin. The lipofuscin is the normal age-related aggregations. Although those kind of aggregates possess the autofluorescent property, but the appearance of them are quite different. The autofluorescent aggregate is larger and denser. However, the lipofuscin is like a granular-like dot-like structure, just like a little dots. Yeah. And in the patient frontal cortex, in the neurons and microglia cell, we can observe that in the FTD patient, there are lots of autofluorescent aggregates in the neurons and the microglia cell compared with the control groups. And about the average coverage of the autofluorescent aggregates, it mostly happens in the FTD patient, and also, but the lipofuscin usually will happen in the normal human beings, which is the control group. So we can easily distinguish them with each other. So now I'm going to talk about the details about the story of this paper. The first result shows that the author created the artificially truncated CAM2B mutant mice and observed that there are lots of larger and dark aggregates here in the truncated mutant mice, which is the autofluorescent aggregates. And a small arrow indicates the lipofuscin, which is more lighter and dot-like structures. Not only in the thalamus, but also in the cortex show the same trends. In the truncated mutant form, here are the autofluorescent aggregates, but in the CAM2B wild-type transgenic mice, there is just the lipofuscins. And then the author wants to distinguish the autofluorescent aggregates from the P62 inclusion, which I just mentioned before, which is mostly can be observed in lots of dementia diseases patients. So he used the P62 antibody to recognize the P62 inclusion and used the beta-3 tubulin to recognize the neurons. So in the merged forms, we can see a less co-localization between the P62 and the autofluorescence. In the, not only in the thalamus but also in the cortex, the red one is the P62 inclusion, and the white one is the truncated mutation of the CAM2B. Also, the author defined the Peterson correlation coefficient values. And in both cortex and thalamus, it shows a very low value, which means there are less correlation between the P62 inclusions and the autofluorescent aggregates. So there are totally two different kinds of proteins. And thirdly, the author wants to identify the specific location of the autofluorescent aggregates in the CAM2B mutation. So he used the marker to mark the neurons and microglia cell respectively with the tubulin and the IBA1. And in the merged form, there are highly colocalization between the autofluorescent and those cell types. Especially in the microglia cell, there are nearly 90% of microglia cell contain this kind of autofluorescent aggregates. However, in another hand, in the P62 inclusion, mostly exists in the oligodendrocytes process, which is marked by the CA2. So you can see that the red dots and green dots have several colocalization, and the I95 is used to mark the exome process. So we can, it further suggests that the P62 have different expression patterns and location with the autofluorescent aggregates. So there are two different kind of proteins. In more clinical respect, some doctors have shown that the P62 inclusion predominantly occurs in the hippocampus instead of in the frontal cortex. So I think it's quite paradoxically because the neurodegenerating area of the FTD patient is usually in the frontal cortex. However, the autofluorescent aggregates are usually located in the patient's frontal cortex. So it might be a more evidential biomarker to detect the FTD. And the last result, the author would like to ask which kind of mechanism will cause the neuronal loss in the patient frontal cortex. So under the electron microscope, the darker part is the autofluorescent aggregates, and the NU is the nucleus of the neurons. And there are two different structures, which is the multivesicular body and the lamina membrane rows. Those structures are frequently exist in the late endosomal process. So it shows that the autofluorescent aggregate is highly associated with the endolysosomal process. To be more evidential, the author utilized the antibody to mark the late endosomal protein, such as the LAM1 and LAM2. you can see there are lots of small black dots, and just nearby the autofluorescence aggregation. So we can see that the autofluorescence aggregate is highly correlated with the lysosomal system. And then this Western blotting shows another interesting result, which is that the CAM2B mutation will impair the endolysosomal process. So there are lots of late endosomal proteins cannot be successfully degraded by the lysosome. So we can compare by the Western blot membranes data and the quantitative data. So the autofluorescence aggregate is not only just colocalized with the endolysosomal system, but also will impair the function of the lysosomal degradation process. And in my last part of my presentation, I will throw several discussions that I think it is quite interesting. The first one is the excitation spectrum of the autofluorescence aggregates. Because in this paper, the author just shows that says that the autofluorescence can be excited by the whole spectrum, but I think it is quite ambiguous. It needs to be narrowed down to a very specific spectrum for the excitation. So we may get a higher intensity and exclude some extra background noise. And another question is about what is the P62 inclusion roles placed in the FTD patient, because it's usually located in the hippocampus instead of the frontal temporal lobe, but the neurodegenerative area are in the frontal temporal lobe. So I wonder about whether this inclusion have another kind of function instead of killing the neurons in the patient's frontal cortex. And the third one is, I'm wondering about is the lipofuscin is a precursor of the autofluorescent aggregates. Because the appearance of the lipofuscin is smaller than the autofluorescent aggregates, so it may share a similar pathway. Otherwise, there are two totally different pathways to form those different kind of aggregation. So I think those questions can be do more in the future neuroscience field. And lastly, I would like to sum up my paper. The author utilized the CAM2B truncated mutant mice and found lots of autofluorescent aggregates in both the thalamus and the cortex. And their existing patterns are quite different with the P62 inclusion in the CAM2B truncated mutant mice, which shows that there are two different kinds of proteins. And the third one is the autofluorescent aggregates might impair the endolysosomal system. Yeah, and highly associated with the endolysosomal system. Oh. And lastly, I'd like to say that I think the autofluorescent aggregates are very promising biomarkers because it only relies on the natural property of the protein instead of stain the protein with the artificial antibody. So it will be more straightforward. And I think if this technique can be improved in the future, it can help lots of dementia patients and to improve their life qualities. And that's all of my presentation. Thank you for listening.
好，现在是教授提问时间。
The author mentioned anything about the component of this autofluorescence?
The component.
Besides P62.
I think
they propose anything?
This paper only shows that there are lots of autofluorescence, but it didn't mention the component. But I have searched the autofluorescent protein on the Wikipedia. There are several amino acids will possess this kind of property. But I think this question should be further discussed and do the research in his lab, because he didn't mention more about the component of the.
Why not go intron 5?
Intron 5. Oh, because the intron 5 is the C-terminal trunk. It's in the C terminal, and the author says that the functional part is near the C terminal of the CAM2B. So he tried to knock out the C terminal to form the C terminal truncated CAM2B proteins. So to make it loss of function.
Do you think this truncation will mimic what's happened in the genetic mutation occur in the FTD patients? Is there any patient detected with this intron 5 deletion?
Yeah, in my previous slide and in this one, in the paper it mentioned about those images are imaged from the patient's frontal cortex, and those patients have been diagnosed with the intron 5 truncated form.
So this mutation is intron 5?
Yeah, so that's why the author will truncate the C-terminal.
This is a follow-up question. When we're reading an article like this, I think it's very critical for us to learn how to ask the next question. So here I would like to challenge you. You were the authors of this article. What kind of approach or research design would you use to reveal the exact identity of the autofluorescence aggregate?
About which kind of the result, I've prepared an extra slide, which is mentioned about this spectral imaging with subsequent linear omising, which is done by using by adjusting the dichroic mirror and the fluorescent filter inside the confocal microscope to split different signal into different light paths. So the different kind of signal cannot mix with. It's impossible to mix with each other. Yeah, because in the data I just showed, the author also used different antibody to mark, such as the microglia and the neurons, and also detect the signal of the autofluorescence, because there are many kind of the signal. So he utilized this kind of techniques to separate different signal into different light paths and detect it by different kind of detectors. So to avoid them to mix up together. So I think this technique can increase the identity of observing the autofluorescent aggregates.
Well, actually, you misunderstood my question because I want to ask what the professor wants to ask you the exact component of this autofluorescent aggregate. You say the author didn't answer the question, so this is what I'm asking you. If you are the researcher, what kind of study will you take to identify the exact component of this autofluorescence protein?
I will search for several review papers about the autofluorescent protein and use the maybe some biochemistry like the Western blotting and use the antibody to recognize it. Is there any kind of candidates can be detected in the autofluorescent aggregates? So it might be the components. So do I answer your question?
Yes.
You need to first have an idea of which protein you are going to test before you do the Western. So you need to guess.
Need to guess.
So Boson's nice talk, and also any other kinds of cells has this autofluorescence protein existing?
In this paper, it just mentioned it mostly predominantly occurs in the neuron and the microglia cells. And however, P62 inclusion is mostly occurs in the oligodendrocyte. So there are two different kind of proteins.
So what is the function of P62 in this process?
The author wants to distinguish between the P62 and the autofluorescent aggregates. And he wants to exclude the possibility of P62 will affect the pathology of the FTD. Yeah. And he also says that the P62 usually locates in the hippocampus of the patient, of the FTD patient, but the FTD patients, their neurons are usually lost in the frontal lobe instead of the hippocampus. So I think there might be another kind of function instead of killing those neurons in the FTD patient.
So is this P62 2 is the protein involved in the whole